Hello everybody, this is Mitosis Part 1. I would like you to open up your books to Chapter 12 and have out your Chapter 12 reading guide so that I can point things out as we go and you can add to your notes. And we're going to also do the concept checks, some of the concept checks together as a part of this video, because I think that will help you to review while I'm not there. So beginning with this slide this is Kim Foglia's PowerPoint. I really like her PowerPoints. I apologize in advance if there's weird transition noises that she adds in, but the rest of her PowerPoints are really great. So biology is the only subject in which multiplication is the same thing as division. Hardy har 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 har. Okay. All right, so we're going to start by talking about the cell cycle. And that will include cell growth and cell division, mitosis. So the entire life cycle of a cell includes both all of its growth and its division. And of course, during the growth times, we're also doing things like making proteins and making phospholipids and all of that as well. And what we're showing you here is a plant cell undergoing mitosis and an animal cell undergoing mitosis. And you'll be doing some of this in the lab. You probably remember doing a version of that lab in freshman biology too. All right, so where does it all begin? We started as a single cell, smaller than a period at the end of a sentence, teeny weeny little beings that we all begin as. And now look at you. And here's some pictures of teenagers. So it's kind of hard to imagine going from being a single cell to lots and lots of cells. So you go from being this tiny little being to a ginormous being. These pictures are not coming out, which is fine. All right, so how do you go from being a single cell to a fully grown human being? So it starts with a single cell, which replicates and divides again, and divides again. So the original fertilized egg has to divide, and divide, and divide, and divide. So why do cells divide? What do you think? So reproduction. So if you are a single-celled organism, you would divide as your strategy for reproducing and increasing your population. So there's a little amoeba, okay, dividing in order to make more of itself. Okay, so that's a part of asexual reproduction for single-celled organisms. And on the other hand, cells divide for growth. So to go from a fertilized egg to a multi-celled organism, that's another good reason to divide. And another reason to divide is for repair and renewal. Cells can't last forever. All right, so these examples are organisms which, when they lose a significant part of themselves, can regrow it, which is completely cool. So it can replace cells that die from normal wear and tear or from injury. And this is red blood cells, which definitely wear out and need to be replaced. So how do we make new cells? We're going to go over some of the anatomy of a cell that's involved in this process. So the nucleus, of course, is where the chromosomes are, the DNA. Okay, the cytoskeleton is involved, so providing a structure for how all of this is going to go down. The centrioles in animals, microtubule spindle fibers. Okay, so we're going to go through the structure of the nucleus and how the DNA is arranged. Okay, the nucleus is there to protect the DNA so that the rest of the cellular structures and enzymes and other macromolecules do not affect the DNA. So it's safe within the nuclear membrane. All right, so there's a nuclear envelope, a membrane, a double membrane, around the outside of the nucleus that has nuclear pores in it. Okay, it's a double membrane, and the membranes fuse in spots to create pores, and by pores I mean little holes, little tiny holes. Okay, and you can see in this picture the membrane shown in green with the little holes here, and the nucleolus in the middle. Okay, so the pores allow large macromolecules to pass through. So the question is, what kind of molecules need to pass through there? Well, you probably remember, like, how do we get the information from DNA out to the rest of the cell? And that's when we're going to be talking about transcription and translation. So we need to get messenger RNA out of the nucleus to go out and find the ribosome. So these holes are large enough to allow messenger RNA to pass through, but DNA cannot pass through them. So this giving you just a little bit more detail about the anatomy of the nucleus in both a drawing and the scanning electron microscope here to show you what the structure really looks like. So this is an awesome picture of these tiny pores, which are really cool. And this is giving you kind of a cross-section view of the membrane, the inside of the nucleus, the cytoplasm, and the pore. All right, and here's the endomembrane system, the rough endoplasmic reticulum attaching to the outside of the nucleus. The cytoskeleton is going to be providing the structural support, maintaining the shape of a cell, and provides a way for the organelles to stay in one place, to, if they're meant to stay right in one spot, with certain protein fibers, which are microfilaments, intermediate filaments, and microtubules. And we're going to see how those have a role in mitosis in a second. The other part of the function of the cytoskeleton is motility, meaning movement, how a cell moves using, especially if you're a single-celled organism, using cilia and flagella to move. And the other function of the cytoskeleton is regulation, organizing the structures and activities of the cell. So we're going to show you a detailed picture here to give you a way to picture this. The cytoskeleton is on page 112 and 113 in your textbook. When we tested you on chapter 6, we really didn't go into this because we knew we would be getting more involved in it for this unit. So if you are following along with your textbook, I would look at page 112 and 113 to better visualize the cytoskeleton. So this picture is showing you the elements of the cytoskeleton and how they're interacting with the other organelles. So this is showing you the microfilaments which are involved in maintaining the cell shape. And they're also involved in cell division in the formation of a cleavage furrow, which we'll talk about in a little bit. So the microfilaments are right here, and they're in the chart on page 113. Those are the actin filaments. Microfilaments are also known as actin filaments because it's two strands of actin, which is a protein. So, so that's that one. Then we have the microtubules here, okay, which are the blue tube-looking things, the microtubules, which are tubulin polymers, that they're made of tubulin. They also maintain the cell shape. They can be involved in cell motility. They're also involved in the way that chromosomes move during cell division and also how organelles move and are supported in the cell. So that's microtubules. There's a really interesting picture on page 112 of your book showing you how microtubules, which are little tubes of proteins that have formed in a spiral to create a tube, and showing you a vesicle walking along that tube. And we saw that when we watched the Inner Life of the Cell video. We saw how it looked like little feet walking along a tube, and that's how vesicles can move along from the inner parts of the cell to the outside of the cell, or vice versa. Okay, we also have intermediate filaments, which are here in the green, and those are a little bit thicker and more fibrous, and they're also involved, of course, in maintaining the cell shape. They hold the nucleus and other organelles in place, and they form the nuclear lamina. So for cell division, the other parts are a little bit more important, but they all play a role. So if you didn't learn about the cytoskeleton in Chapter 6, you're going to want to maybe make yourself a couple flashcards for the 3 types of cytoskeleton elements: the microfilaments, the intermediate filaments, and the microtubules. Okay. So centrioles, okay, in a lot of cells, microtubules grow out from a centrosome, an area near the nucleus that's like the microtubule organizing center. And these microtubules are resisting like being smushed. So they're like holding the structure. And within the centrosome of animal cells are a pair of centrioles. They're made of microtubules arranged in a ring, and before a cell can divide, the centrioles replicate. So centrioles organize microtubule assembly, and they're in animal cells. So it's part of cell division. It's, as I said, in animal cells, a pair of centrioles organizing the microtubules and the spindle fibers, which are essential for mitosis, and we'll talk about those in a couple minutes. And they guide the chromosomes in mitosis. So those are the parts of the cell that we're going to be talking about in mitosis are the parts of the cytoskeleton, the centrioles and centrosomes, the nucleus. What did I forget? Oh, and we'll talk about the anatomy of the chromosomes in a few minutes. So that's the end of part 1 of mitosis, and you can start part 2.